Hi everyone, I am Dr. Susmita and in this class I am going to discuss about the antiarrhythmic drugs. Under this topic I will discuss about the classification and the pharmacology of the drugs which are used in the treatment of arrhythmia. Before starting with the pharmacology, let me explain you about the cardiac conduction, cardiac physiology and the pathophysiology of the arrhythmia which will help you to understand the mechanism of action in the better way. So let's start with the cardiac conduction. So the heart is having the two cells. There are conducting cells and the contractile cells. Conducting cells are the cells which generate the electrical impulses itself, and contractile cells are the cells which receives the electrical impulses. So in the conducting cells include the sinoatrial node, atrioventricular node, the bundle of His and the Purkinje fibers. So what happens is in the first SA node, the SA node, it is a group of a cell which is found at the right atrium which is close to the junction with the superior vena cava. This SA node have a function of intrinsic pacemaker cell. That means these nodes generate the electrical impulse itself. It does not need any neuronal activity to generate the electrical impulse. As these SA nodes generate the electrical impulse, those impulses are transmitted to the right and the left atrium, and those electrical impulses stimulate the atrial myocardium to contract. The contraction of this atrial myocardium are relatively very fast. Now the next one is atrioventricular node. These cells are situated in the atrioventricular septum which are just above the coronary sinus ostium, and this atrioventricular node receives the electrical impulse from the atria and it transmits the electrical impulse from atria to ventricular, and the process of transmitting this electrical impulse from atria to ventricular is relatively slow, as is a slower conduction process. Therefore it allows the ventricles to have a maximum filling before the contraction. Now the next one is the bundle of His. The bundle of His, it is a collection of the heart muscle cell which is specialized for electrical conduction, and this cell receives the impulse from the AV node. As it receives the impulse from the AV node, then it transmits this impulse to the left and the right bundle branches which travel down to the intraventricular septum, and from those septum it travels to the both right and left ventriculars respectively, and from there those impulses are then moved to the Purkinje fibers. Now these Purkinje fibers are situated in the subendocardium region of the heart, and they transmit the wave of electricity to the ventricular myocardium, causing the ventricular contraction. So both of these cells are helping in the cardiac conduction and the contraction. So what we study over here is that SA node, which is a major pacemaker cell, is having an intrinsic activities, that is, it generates the electrical impulse itself. As it generates the electrical impulse itself, it transmits the electrical impulse to the atrium, and from the atria it moves to the atrioventricular node. From the atrioventricular node, the electrical impulse moves to the bundle of His, and from there it moves to the Purkinje fibers, causing the conduction and the ventricular contraction. So here SA node is the major pacemaker cell, and the other, that is AV node, bundle of His, and the Purkinje fibers are the latent pacemaker cell. So I hope you understood about the cardiac conduction till here. Now let's see about the cardiac physiology. So for the cardiac physiology, we will first see about the SA or AV nodal action potential. So here we have a SA and AB nodal cell. So in this cell we have a sodium channel, potassium channel, T-type calcium channel and L-type calcium channel. So as I explained you that SA node is generating the electrical impulse itself. So as it generates the electrical impulse itself, it activates the sodium channel, which makes the sodium channels open. As the sodium channels get open, sodiums enter into the cell. As the sodium enter into the cell, it makes the cells positive. As the cells become positive, then it will move from resting membrane potential to the threshold potential. And this phase of moving from resting to the threshold potential level, it is of phase 4. Now, this positiveness of this cell, what it does is it will activate these calcium channels. When this activates the calcium channels, it makes the calcium channels to open. As these calcium channels get open, the sodium channels get closed and the calcium enter into the cell. As the calcium enter into the cell, the cells became more positive, and this positiveness of the cell will cause this cell to move from threshold potential to the peak level. As it reaches to the peak level, which is the phase 0, then the potassium channel get activated and it gets open. As it open, then potassium from this cell move out of this cell, making this cell negative. As these cells start to turn out to be negative, then it will move from peak to the resting membrane potential, and this phase is called as the phase 3. Now it has reached to the resting membrane potential. This resting membrane potential is maintained for a while with the help of the pump, that is the sodium potassium ATPase pump. So what this sodium potassium ATPase pump do? So these pumps, they make sodium to move out of the cell and potassium to move inside the cell, and these are maintained for a while, maintaining the resting membrane potential. So I hope you understood this process. So what happening here is, when sodium channel is getting open, that is the phase 4, and when calcium channels open, that is the phase 0, where the actual depolarization happens, and when potassium channels open, that phase is phase 3. Now the next. Here we have an arterial or ventricular cell, and in this we're gonna see the arterial and ventricular cell action potential. So from the pacemaker, the electrical impulses will move into the cell via gap junction. As the electrical impulse move into the cell, the sodium channels get open, and these sodium channels will cause the rapid entry of the sodium ion inside the cell, making the cell positive. Due to the rapid entry of this sodium ion and making the cell positive, the cell from the resting membrane will directly move to the peak phase, and this phase is called as the phase 0. Now this cell has moved to the peak phase, so what it will do is it will activate the calcium and potassium channel, causing it to open. As these channels get opens, then potassium from inside the cell will move out, making the cell negative. As the cells turn negative, there will be a slight decrease into the potential. Now, here as the potassium is moving out, but the calcium channels are also open, which will make the calciums to enter into the cell, making the cell positive. As the cells are being negative in one side, but the other side, due to calcium, it is also being positive. So this voltage is maintained for a while, and this action potential is maintained for a while, causing it to be forming a plateau phase, and then this plateau phase will form, which is the phase 2. Now the positive, now what happens is the calcium which is present inside this cell and has been positive, it will activate the voltage gated channel, that is the sarcoplasmic reticulum. As it activates this reticulum, what it does, it will stimulate the cell to release the calcium out of the cell. As the calcium moves out of this cell, then the relaxation process will happen, because when calcium are inside the cell, it is contracting, and when calcium are moving out of the cell, then it's relaxing, because as it move out of the cell, the positiveness from inside the cell will move out, and here potassium channel is still open, so potassium is still going out, due to which the negative in this cell will increase, causing the relaxation, and from the plateau phase it will again go back to the resting membrane potential, and this phase is called phase 3. And this resting membrane potential are maintained for a while with the help of a pump, that is a sodium potassium ATPase pump. What this pump is do, it will move sodium out of the cell and potassium into the cell. So that's how the myocardium cell maintain the action potential and causes the depolarization and repolarization. So what is depolarization? When depolarization is the process when the electrical impulses are more positive and less negative in the cell, and it happens when there will be a contraction of these cells. I hope you understood the cardiac physiology. Now let's move to the pathophysiology. So tell me, what is arrhythmia? So arrhythmia is the condition where there will be a irregular heartbeat or abnormal heartbeat, and it is divided into bradyarrhythmia, supraventricular tachyarrhythmia, and ventricular tachyarrhythmia. Bradyarrhythmias are also called a bradycardia because these are caused due to the dysfunctional automaticity in the pacemaker cells. And supraventricular tachyarrhythmia are also called tachycardia, and it is caused due to the impulses which are originated from the atria. And ventricular tachyarrhythmia are the arrhythmia which are caused due to the impulse which are originated from the ventricles. Now let's see what is causing the arrhythmia. So arrhythmias are caused when there will be either abnormal impulse formation or abnormal impulse transmission, which means the transmission of impulse from one cell to the other cell. And the abnormal impulse formation happens when there will be an increase in the abnormal automaticity. So what is automaticity? Automaticity is the ability to depolarize the cell. So here the SA node is the primary pacemaker of the heart, which directs the heart rhythm during the normal circumstances and makes the heart to rhythm, and that rhythm is called as a sinus rhythm, which generally happens during the normal conditions. So what happens as there will be an increase into the automaticity, as the SA nodes are the primary pacemaker cell and it will cover the one action potential. But before it covers the one action potential, the other action potential starts to form that will lead to the increase into the automaticity, and that increased automaticity will cause the increase into the heart rate, leading to the condition of cardiac arrest. And this will only happen if SA node is destroyed, but which is very rare. And normally when automaticity increases, when we do exercise, when we do the exercise, our cardiac output will be increased. As the cardiac output is increased, the automaticity will be increased. That's why it's very normal during the exercise. But when we are in the resting phase, when we are not doing any physical activity, even that time, if there is an increase into the automaticity, then it will cause a sinus tachycardia and also the ventricular tachycardia. Ventricular tachycardia is caused when there will be a abnormal automaticity into the Purkinje fiber, which happens due to any necrosis in the myocardium region. Now the next is triggered activity. So when does this triggered activity happens? So triggered activity happens it either during the depolarization phase or after the depolarization phase. So what happens? So normally how our heart beats. So normally our heart beats when it has an action potential. First it depolarizes and then it repolarizes. During then after when it depolarizes or after the depolarization, there will be some extra beat, and sometimes that extra beat are so high which trigger the action potential to depolarizes the cell and makes the cells to reach the threshold, which produces the premature action potential, and that extra heartbeat is also called a extrasystole, and that triggering activity will lead to the arrhythmic condition. And this triggered activity usually happens in these two phases, that is the first is early after depolarization and the second is late after depolarization. So in the early after depolarization, it happens during the repolarization process, that is in the event in the early the after just after the depolarization. In the early phases there will be extra beat which will cause the condition like bradycardia, hypokalemia, hypoxia, acidosis, hypocalcemia and some other effects. And the next triggered will be late after depolarization, which is it happens after the repolarization process, and in this process are usually seen when digoxins are overdosed or there are strong asympathetic stimulation. So now I hope you understood about the abnormal impulse formation. Now the next is abnormal impulse conduction. So here we will see like how this abnormal impulse transmission occur. So normally what happens is, as the depolarizing impulse spreads quickly and evenly through the heart muscle, it moves like a smooth wave, like it's spreading across. But what goes wrong is when these impulses are moving, they come to the areas which are not in excitable form or where the conduction are not proper. That means that area is blocked, due to which the impulse get blocked in direction. But what this impulse do, as it blocked in one direction, but it is still pass around that blocked area and find the another route to pass the impulse, and that another route, which is also called the re-entry, will form a loop, and that through that loop the impulse will move, then again it return back to the tissue. And this whole process will sometimes happen in the rapid format, and this rapid form will cause the abnormal impulse conduction, causing the arrhythmia. So it is also happening in the two phase. The first one, it happens during the anatomical re-entry. So what happens in this is, in this phase the obstacles are fixed. So here the loop through which the impulse are passing, it goes around the fixed obstacles, for example like atrial flutter circling the tricuspid valve, and these impulses are for the longer period of a time, which will lead to the long tachycardias. And the next is functional re-entry. Here these physical obstacles are not fixed. So the loops form, and here the loops are formed because of the local electrical differences. That means the circuits may be either small or maybe unstable. So due to which the impulse could not pass properly and caused atrial fibrillation. So this is all about the pathophysiology of arrhythmia. I hope you understood this pathophysiology of arrhythmia. So in this class, what we studied is we covered about the cardiac conduction, where we come to know that the SA nodes are the major pacemaker cell and which helps in the maintenance of the heart rhythm. Then we studied about the cardiac physiology, where we come to know about the action potential reaction of the cardiac cells. Then we studied about the arrhythmias and its pathophysiology. So I hope you have understood till here, because this will really help you to understand the mechanism of action of antiarrhythmic drugs. Now I will wind up this class, and in the next class I will explain you about the classification and the pharmacology of the antiarrhythmic drugs. Thank you.